Good day everyone. Today we are going to present you Experiment 5, Cellulose Degradation and Glucose Analysis. We are from Group 5. My name is Lim Chu Tian and my group member No Anissa, Chu Hui Ning and Yong Wen Chun. We are supervised by Dr. Era and we are from Section 2. Objective: to understand and explain the method of cellulose degradation and analysis of glucose. To conduct and explain the glucose analysis using dinitrocyclic cyclic assay. Now I pass the presentation to Hui Ning.
Hi Dr. Era and everyone, I'm Hui Ning. Now I will talk about the theory in this experiment. Cellulose is the most abundant biopolymer on Earth and it usually used as a reinforcement material. For example, we can find the cellulose in the cell wall of plants, which help the plants to stay upright. This organic compound has the empirical formula, which is the C6H10O5N, and cellulose is composed of a long chain of at least 500 glucose molecules. And this is the structure of the cellulose. Besides, the cellulose is secreted by specific species of bacteria to form biofilm. Glucose is the primary carbon source for microbial growth, so for presenting a cellulose degrading activity, cellulose biomass could be used as a potential carbon source, as D-glucose is its building block. So this process we call it as cellulose degradation or hydrolysis reaction, which is a process where the cellulose converts into the glucose unit. After cellulose degradation, glucose will be produced. Glucose is one of a group of carbohydrate known as 6-carbon monosaccharide, which cannot further hydrolyze. The molecular formula of the glucose is C6H12O6. It is the source of energy in the cell function, and the regulation of its metabolism has great importance in fermentation and in gluconeogenesis. However, the presence of the glucose is difficult to identify because of its colorless appearance. But the carbonyl group in the monosaccharide are able to undergo the oxidation, so few oxidizing agent can be used to convert the colorless glucose into the colored compound. Hence, we can undergo the glucose analysis to detect the presence of the glucose. For detect the glucose, one of the common reagents to detect the presence of glucose is 3,5-dinitrocyclic cyclic acid, which is known as DNS. It can analyze the glucose content, and it is introduced by Sumner in 1921 for the estimation of sugar in normal and diabetic urine. DNS can detect the presence of free carbonyl C double bond O group of reducing sugar, and this involves the oxidation of the aldehyde functional group which is present. For example, the glucose and ketone functional group in fructose. At the same time, 3,5-dinitrocyclic cyclic acid DNS is reduced to 3-amino-5-nitrocyclic cyclic acid under alkaline condition. When 3,5-dinitrocyclic cyclic acid, whose yellow color is being boiled, it will react with reducing saccharide by boiling and change into 3-amino-5-nitrocyclic cyclic acid, whose color is orange-red. The effect of DNS to glucose are different for each concentration of the glucose. The level of the cellulose degradation of microbial culture can be determined by spectrophotometric analytical method. This method is based upon the color which forms when D-glucose reduces the 3,5-dinitrocyclic cyclic acid to 3-amino-5-nitrocyclic cyclic acid due to its strong absorbance at 540 nanometer. In this experiment, we use the ultraviolet visible spectrophotometer. This device can be used to measure the absorbance, which also known as the optical density. Absorbance is the quantity of the light absorbed by a solution. The value on the screen, which shown on the slide, can indicate the absorbance of the sample liquid. However, the absorbance value which measured by the spectrophotometer does not describe the concentration of glucose. So, the concentration of glucose can be determined by using the standard curve. The standard curve can be constructed by measuring the absorbance of a solution with a known concentration of the substance being measured. The standard calibration curve in this experiment will label the x-axis as the concentration of glucose, while y-axis as the optical density value. From the calibration curve, we can determine the concentration of glucose of the sample. So that's all from me. I will pass to Chiu Tian to continue talk about how to conduct the experiment. Thank you.
Thank you, Hui Ning. Now I will continue the presentation on apparatus and material. For the first one, microbial solution, which is overnight culture. And second, dinitrosalicylic cyclic acid solution, sterile distilled water, universal bottles, glass pipette and pipetter. And the last one, D-glucose solution with concentration 1 mg/ml. For the procedure, the standard curve for glucose analysis will conduct. For the first step, 5 dilution of the D-glucose solution were made. The acetal buffer provided was used in all dilution. That was the glucose standard. The dilution calculation was shown in the report. And then 1 ml was taken from each dilution into a new bottle, and then 1 ml of BNS reagent was added. All tubes were boiled for exactly 5 minutes in 90-degree water bath and cooled before adding 3 ml of distilled water. And the last step, OD540 was used to measure the glucose content in all tubes in the UV spectrophotometer. For cellulose degradation, the first step, 4 different cellulose degradation trials were given, which were conducted the day before the experiment. They were put in 2 ml centrifuge tube and were centrifuged for 5 minutes at the highest speed. And the last step, the solution part was pipetted and the reducing sugar content was analyzed. That's all the presentation from us. Thank you.